Then types of cells. In the living organisms, basically there are two types of cells. One is prokaryotic cells and eukaryotic cells. So these are the cells have been divided into two types based on some specific characters and based on presence and absence of some structure, cell organelles or the different aspects of the cell. So like if you take the prokaryotic cells, so generally these are the one which lack a complete nucleus or definite nucleus. Here what is a complete or definite nucleus means the nucleus which contains nuclear membrane, nucleoplasm, chromatin material and also nucleolus, that is called as a complete or definite nucleus. So here eukaryotic cells contain complete or definite nucleus. The one which does not have such a complete or definite nucleus are called as prokaryotic cells. But how does the nucleus is represented in the prokaryotes? Actually here in prokaryotes the nucleus is represented only with the naked circular single double stranded DNA molecule. So no nuclear membrane, no nucleolus and no nucleoplasm in it. So there is a name given to this nucleus of prokaryotic cells, that is called as genophore or prochromosome or nucleoid. So that is the basic difference that we find here, and apart from that one we know that chromosome should contain the DNA along with the histone molecules, but here the histone molecules are also absent in the prokaryotic cells. So likewise some cell organelles are also absent here, like double membrane structures, and also some single membrane structures are absent here, like mitochondria, plastids, that is nothing but chloroplasts, along with endoplasmic reticulum, Golgi complex, microbodies, microtubules, lysosomes. Such type of cell organelles are absent in the prokaryotic cells. Now let us discuss the basic features that are present in a prokaryotic cell, where it is differing from the eukaryotic cell. In the evolutionary aspect, the prokaryotes are considered as a primitive one, where in the universe we said that bacteria are the primitive structures which are formed in the universe. In the course of their changes, they are converted into the eukaryotic organisms, and whatever the much complex living organisms that we are observing here, they have been created, they have been formed from the modification of the prokaryotic organisms. So here the prokaryotic cells are basically seen in bacteria and cyanobacteria like examples. So here the prokaryotic cells contains cell wall, and this cell wall is not a usual cell wall that we can see in a eukaryotic organism, and apart from this one it also contains the plasma membrane inside the cell wall. So it is of course as like that of eukaryotic cell, it is also lipoproteinaceous, where it is made up of lipids and proteins. Then inside this one the protoplasm is present. The protoplasm, which is actually we know that when we say it has protoplasm, it should contain the cytoplasm along with a nucleus. But here as I said that the nucleus is represented with a naked structure called as a nucleoid along with the cytoplasmic content. In the cytoplasm, where mitochondria, endoplasmic reticulum, Golgi complex, plastids, particularly plastids are present in the plants, lysosomes, microbodies, microtubules, etc. Or we can say that single membrane units and also the double membrane units. So double membrane units are mitochondria and plastids. That means they are covered with or they are surrounded by two unit membranes of protective structures. Single unit membrane structures are endoplasmic reticulum, Golgi complex, lysosomes, microbodies, microtubules, etc. All these structures are also absent in the prokaryotic cells. Then there are some photosynthetic or the autotrophic prokaryotes are there. So when you say that an organism is autotroph or autotroph or an photosynthetic, it should contain the chloroplast along with the pigments. So actually here in the photosynthetic prokaryotes, they contain thylakoid structures. Actually thylakoid should present in the chloroplast. As the chloroplasts are absent, the thylakoid structures are present in cytoplasm only. Then there are some aerobic prokaryotes are there. These in the aerobic prokaryotes, where of course the mitochondria are absent. Instead of this one, the membrane structures, instead of the membrane mitochondrial structures, they contain some special structures which are concerned with the respiration. Some of the prokaryotic cells contain some special type of vacuoles. They does not have the normal vacuoles that are observed in the eukaryotic cells. So these are a specialized one which are concerned with buoyancy or osmoregulation. So where they will not allow the passage of water, but only the passage of gases is allowed. Then coming to the sexual reproduction. Generally sexual reproduction is absent in this one because there is no gamete formation as like that of normal eukaryotic organisms. But here the transfer of genetical material from the parent generation to the offspring generation will be taking place. Then other aspects related to the prokaryotes, the histone molecules are absent. Histone proteins, they should be present along with the DNA inside the chromosome. That histone proteins are absent here, but they have the non-histone molecules. So likewise there is a lot of variation that can be observed in the prokaryotic cell when compared to the eukaryotes. But when it is coming to the non-cellular organelles like in ribosomes, so here prokaryotes contains 70s type of ribosomes. 70s type of ribosomes are present in this one, and each ribosome contains two unequal subunits, the larger subunit and the smaller subunit. Larger subunit is made up of 50s and smaller subunit is made up of 30s. So these ribosomes are concerned with the protein synthesis. So these are called as protein structures or the working benches of the proteins which are concerned with the synthesis of proteins in the prokaryotic organisms. But in eukaryotes it is of 80s type where they are concerned with the protein synthesis. Let us take the typical example of prokaryotes, that is bacteria. If we observe the structure of bacteria, so we know that in general, generally in living organisms, the outermost limiting membrane should be cell wall. But here outside the cell wall, they contain some other additional layers. So that additional layer is called as a capsule. So this dotted structure, actually it is a structure which is present outside the cell wall called as capsule. Actually glycocalyx we call it as. It may exist in two forms. One is a capsule, and the glycocalyx will exist in either form of capsule or mucilaginous layer. When the glycocalyx exists in a tough and thick sheet, so it is called as capsule. Whereas if it is in the loose sheath like structure, we call it as a mucilage layer. Then inside the glycocalyx, of course here these are concerned with the protection of the cell from exit of water or desiccation. Then inside this one, this is a cell wall. This cell wall is not an cellulosic cell wall as a typical normal plant cell, and this is made up of peptidoglycans. Otherwise these are called as mucopeptides, muramic acid or murines. Then inside the cell wall we can see the presence of plasma membrane. So plasma membrane of the bacteria is a normal plasma membrane that can be seen in a typical eukaryotic organism. Which is made up of lipoproteins. And in the plasma membrane, here and there we can see some inner foldings like this. These inner folding like structures are called as mesosomes. So mesosomes are concerned with various functions. So they are the structures which are helping in replication, and also to some extent they consider these mesosomes are having the respiratory enzymes. Then in the cytoplasm we can see the presence of the genophore, that is nucleoid is present, which is represented with a single double stranded naked DNA molecule. And sometimes in some of the bacteria, apart from the nucleoid, the chromosomal DNA part, some additional DNA is present, that is called as this extra chromosomal DNA is called as plasmid. Called as plasmid. It is also present in the cytoplasm on one side. So what is the significance of this plasmid? Due to presence of plasmid, the bacteria will acquire some new characters like resistance to the antibiotics, like antibiotics like streptomycin and ampicillin. So plasmid is the one which is made up of few number of genes, and plasmid is used as a vector in the genetic engineering process. And apart from this one, the remaining part is called as a cytoplasm, which is a normal jelly like substance. In this the cell organelles, as we are saying, mitochondria, endoplasmic reticulum, Golgi complex, microbodies, microtubules, all such structures are absent. But here and there we can see some group of structures which are present in the form of a chain. We call this type of structures as polysomes or polyribosomes. That is, the ribosomes, smaller and larger subunit of ribosomes are binded with the mRNA molecule. So we call these structures as polyribosomes or polysomes. Likewise, lipid molecules and other protein molecules can also be seen inside the cytoplasm of the bacterial cell. And some bacteria which are aquatic in the nature, they contain a special vacuole that is called as a gas vacuole. So this gas vacuole is a membranous structure which will not allow the water passage, whereas it will allow the movement of gases through it. So it is concerned with the buoyancy and little amount osmoregulation also. Then these bacteria or prokaryotes contain some special structures which are called as inclusion bodies. Which are concerned with the storage of the materials like phosphate granules and sulfur granules. Like this type of structures are there which are concerned with the storage of these materials. Here there are many types of inclusions are present which are inorganic and dead things. So here they are Synophycean granules, volutin granules or phosphate granules. In the same way, glycogen granules and Synophycean, like different types of non-living inclusions are present inside the cell. Apart from this one, they have the gas vacuoles also. Then chromatophores. There are some photosynthetic bacteria which contains lamella like structures, like in green bacteria. So here they contain the typical membrane structures. Apart from that one, they contain the thylakoid membranes which are having the which are having the photosynthetic pigments called as bacteriochlorophylls. which are concerned with the photosynthetic function. Then mesosomes, which are the inner foldings extended into the cytoplasm of the bacteria. So these are having lamellae, made up of lamellae, vesicles and tubules. So these are concerned with various functions related to the bacteria cell. So these are concerned with increase in the surface area, and also the enzymatic activity. So here replication of DNA and also the transfer of DNA to the daughter bacteria. Then it is also concerned with the secretion. So like various functions are there, replication and respiration. So like various functions are there related to the mesosomes in the bacterial cell. Then the type of ribosomes which are present here, as we have discussed, as like in the normal prokaryotic cell, they contain 78 type of ribosomes with a larger subunit and a smaller subunit, which are concerned with protein synthesis. And these ribosomes may be present freely in the cytoplasm, or sometimes they may be formed into a chain. So these structures are called as polyribosomes. When they form the polyribosomes, they are concerned with the synthesis of proteins. These are membraneless cell organelles. Then the reserve food materials which are present in the bacteria. The reserve food materials are mostly in the form of glycogen, but in eukaryotes, as we know, it is mostly in the form eukaryotic organisms like plants. So it is mostly in the form of starch, but here it is in the form of glycogen or PHB, poly beta hydroxy butyrates. So these are the form of food materials which are stored in them. So likewise, these are the characters that we can observe in the typical bacteria cell. And some of the bacteria are motile in their nature. So when they are motile, they are having a specialized structures called as a flagella, which are concerned with locomotion. So each flagella will contain basal hook like structure. So basal body and the filament. Basal body is a structure which is present inner side bacterial cell. Hook is the one which is connecting the basal body and the filament, and filament is a long elongated extension coming out of the bacterial cell, and this flagella will show rotation type of movement which will push the water backside. When the water is pushed backside, the bacterial cell will move front. So this type of nature will be there, and flagella is made up of a protein called as flagellin. And this flagella, as we know, if we take the transverse section in a eukaryotic organism, it will be showing 9+2 arrangement, that is 2 central fibrils and the 9 peripheral fibrils are present, but here the central fibrils are absent. Only the 9 peripheral fibrils are present in the transverse section of the flagella. So these are flexible structures which are helping in the locomotion. Apart from flagella, some other specialized structures are present. Those are called as pili or fimbriae. So how they are differing from normal flagella means they are small in size and they are stiff. These are the structures which are concerned with attachment, and the number of pili or fimbriae are more when compared to the number of flagella. Then these are made up of a protein called as pilin or fimbrillin. In these also there is a little modified structures are present. Those are called as sex pili. How does this normal pili are differing from sex pili means? Normal pili have solid nature in the center, that is there is no hollow space, those are solid, and whereas sex pili have hollow space in the center. So pili are concerned with attachment to any substratum, whereas sex pili are concerned with attachment of bacteria to other bacteria during conjugation process. So not all that thing, as it is hollow space is present, this sex pili is used as a conjugation tube during conjugation process. So these are the things which can be seen in the bacterial cell. And here when we discuss about the type of flagellas or the number of flagellas which are present on the bacterial cell. So the bacteria have been divided into various types, that is atrichous. Atrichous means the bacterial cell which is not having any flagella on it. Monotrichous. Mono means single. The bacterial cell contains one flagella. So this is called as monotrichous. Amphitrichous, which contains the flagella on both the poles. One flagella on each pole. Lophotrichous. So the bacteria which contains a group of flagella or a tuft of flagella on one side or on one pole. This is called as Lophotrichous. Cephalotrichous. The bacteria which contains flagella on both the ends like this. So that is a tuft of flagella on both the ends, that is called cephalotrichous. And peritrichous. The bacteria which contains flagella throughout its surface. Many flagella are present throughout the surface on all the sides, so that is called as peritrichous. Likewise, based on the number and arrangement of the flagella, so atrichous, monotrichous, amphitrichous, lophotrichous, cephalotrichous and peritrichous are present. There is a lot of variation in the sizes, shapes and structure of the bacterial cell. So if you see the shapes of bacteria, so basically 4 to 5 types are there, starting with coccus or cocci. Coccus means singular, cocci means plural. So in this these are spherical shaped bacteria are called as cocci or coccus like this. And based on the number of spherical shaped structures, these are monococcus with a single spherical, Diplococcus, 2 spherical shaped bacteria, 2 spherical shaped structures are there in the bacteria. Tetracoccus, 4 spherical shaped structures are there in the bacteria. Streptococcus, where a chain of spherical structures are present. Staphylococcus, with many spherical structures which are irregularly arranged without any specific arrangement. Then Sarcina. Sarcina is the one with which is having 8 spherical structures which are arranged in the form of a cube. 4 can be seen here and the remaining 4 on other side like this. So the cube like arrangement can be seen called as Sarcina. Like this the first one is coccus, and based on the number of spherical shaped structures, so mono to Sarcina are present. Next one is bacilli or bacillus. Bacilli or bacillus means the rod shaped bacteria are called as bacilli or bacillus. So here they appears to be rectangular or rod shaped like this based on the number of cells. So here also monobacillus, only one rod shaped bacteria is present. Diplobacillus, 2 rod shaped structures are present in the bacteria. Then Streptobacillus. Strepto means a chain of rod shaped structures are present like this. So this type of bacillus are there based on the arrangement and the number of rod shaped structures. Then Vibrio, comma shaped bacteria are called as Vibrio. So the best example is Vibrio cholerae. So in this we can see this type of structure. Then Spirillum. Spirillum means spiral shaped bacteria are called as a spirillum, in which the coilings will be there like this. And there are some spirillum which are flexible, so those are called as spirochetes. The flexible one are called as spirochetes. Example is Christispira. Then Begiota is there, which is showing the thread like arrangement of the thread like shape, we can say. Then there is one more special feature is there, that is called as pleomorphism. The pleomorphism means there are some bacteria which will change their shape based on the availability of nutrition. So such type of nature is called as pleomorphism.